empty. I apologize for those that do it every day or read about it every day in the interest of those that don't. So this at the end is a protein. At least this is a way to draw it. Actually in life it is folded, and only the folding, the three-dimensional type folding, allows it to do what it needs to do. It even allows it not to fold or to have different folds. And I really don't want to talk about proteins. I want to talk about the ribosome. So messenger RNA comes to when it comes to the ribosome, or when it's available in the cell, it's a long line. Of course, it has much more complicated structure than my little cartoon here, with 4 different bases. Each of them is called a letter, and triplets of them can code for specific amino acids. So when something like this comes to a ribosome to be translated, first of all, the ribosome has to decide what are the triplets to be translated, because there is a big difference between, for instance, these triplets, if it starts from here, or if one starts from just one nucleotide, one base over. And this is what the ribosome knows to do. It doesn't know to decide which messenger to translate, but it knows to decide where to start and what are the triplets. Ribosome has 2 subunits. I'll come back to it in a second. The small one is the brain. The small one knows where are, or how to assign triplets, and also where to start, where is the initial one. So the initial one has a specific sequence, but it has to find it. And once the specific sequence is found, the first tRNA that brings the amino acid, which is cognate to the first, can be bound. Until now, I drew it just on white background, but actually this happens on the ribosome. So I already said the small subunit knows how to assign the triplets, how to scan for the first one, and allows the tRNA to bind. It makes sure that the binding is correct and that there are as little flaws as possible. So we call it the brain, and the worker, the hands, are the large subunit on which the peptide bond, the bond between the amino acids, will be made. So here is a tRNA, the carrier that is actually quite large, not as large as the ribosome, but quite large, that can find its position for decoding, here, and deposits the amino acid for making a peptide bond. And the way it's done, I have a real quick cartoon for that. The small subunit from the side is rather slim in profile, and people called it a duck in the past, the electron microscopist, with a head and a body and a neck. You'll see from the side that maybe it can remind you of a duck. But anyway, around the neck, the messenger is placed, and this is the first position where the first tRNA will bind, what I just showed you, the initial one. It's called the P position. There are 3 sites, A, P, and E. P is where peptide bond will be made above the peptidyl transferase center that is here, where the amino acid here. There is also an A site for the amino acylated tRNA that will come afterwards, and an exit site here. So once the initial complex is being made, there are factors, I don't show them now. The large subunit can come. There is very nice complementarity between the 2 surfaces, but it's not enough. Conformational changes are happening, and they are making intersubunit bridges. Now the fit should be perfect. The amino acid is now at the P site sitting on top of a tunnel for which the newly born protein will come out. And we think that this tunnel is the way the ribosome protects the newly born protein until it is maybe not large enough to fold, but larger than nothing. Once the initiation complex binds to the large subunit, ribosome can start functioning. Another tRNA comes, it will move here. Peptide bond will be made here real quick. the first one can come out and a new one can come in, and the translocation that follows goes in this direction. So next one will be here a yellow amino acid. Now we have a little movie that we made right in the beginning when we saw the first structures. The movie actually was not made by us, was made by art students from Jerusalem, but it's based on our coordinates in comparison to others. I want just to mention it was done in 2002 when the first structures came. So here is the messenger arriving to the small subunit. There is here a factor, IF3. I actually started with it. And now the large subunit can come with the first amino acid, also brought by a factor. Factors leave, sorry, the first RNA comes. Now the large subunit comes, makes bridges. And this is already the active ribosome. This ribosome can make proteins. More and more tRNAs come, and they stand in line, getting in according to the sequence. Protein is being made here. The ribosome helps. It has a lot of flexibility, getting out, getting in, getting out, getting in, getting out. I want to tell you that this, what we see as hands, for those people that have some experience with flexibility of biological materials, these are 100 nucleotides and at least one protein. On one side there are 4 proteins. It all moves. And I'll come back to the speed in a minute, to the pace in a minute. I just want to show that the protein comes out from the other side and folds either alone or probably more like with the help of chaperones. The whole thing continues until there is a stop codon. Then recycle factors and release factors are replacing the tRNA, and the two subunits can dissociate. So there are many, many ribosomes in each cell. In normal mammalian cells, they can reach 1 million, and in very highly active ones, like the liver, they can reach 6 million ribosomes working at the same time in one cell, all the time, making proteins almost constantly. Even bacteria can reach 80,000 ribosomes working all the time. It means they are really doing a lot of work. And they are doing it very quickly. I promised you to talk about speed or about pace. 5 to 15 bonds per second. 5 in mammalian, 15 in bacteria. I needed a day being a student, a good student. So it's really incredible speed with hardly any mistake. There are mistakes, but the ribosome cannot even proofread them. But most of the mistakes either don't occur or the ribosome proofread. It's about 1 to 1 million. It's a very small number, but from point of view of cell life, it's still significant. So that's what the ribosome does. Even in the 70s, it was already shown that ribosomes are made of 2 subunits, and that many ribosomes work on the same messenger RNA. It means they scan it throughout, and that the proteins come out from the other side. This is an electron microscope from the 70s. I didn't take it. I found it in the literature without any bar and without too much explanation who did what. But even here you can see messenger goes between. Small subunit is bound to the messenger. Maybe here you can see, and protein comes out from the other side. This was all known when we started. What I didn't tell you yet in the ribosome, ribosome is not just two gray bodies. They are made of many components. Small subunit in bacteria has molecular weight of 850. One very large, very long RNA chain of about 1,500 nucleotides and 20 different proteins. Large subunit has 2 long RNAs. One is 2,800, the other about 280, and up to 35 proteins with a molecular weight of 1.5 million. In mammalian, the ribosomes are larger. They can reach 4 million in molecular weight. The structure, though, that structures that are known now are still not of anything else but bacteria, but very complicated. Just to, before I go to the structures, small subunit, I want to repeat, is responsible for initiation, for choosing the messenger RNA decoding and fidelity control, and the large one for peptide bond formation and for getting the newly born protein out. So actually this is the way, what you saw in the movie, there are always 3 tRNAs bound, here small subunit, large subunit. PTC is where peptide bond is being made. This is, if you can see, the messenger RNA. A and P are decoding here, and peptide bond is being made there. And if you want to see it in more detail, although Tanya already showed you, here it is, the large subunit, the small subunit rotating. You see it's a duck? I'll tell you again. Again. A duck? Well, I think it's a Snoopy, but nobody asked me. They gave the names before I went into this field. And the large subunit is called the crown. And maybe now it looks more or less like a crown. See, the king of Sweden doesn't have a crown. It's only the queen and the daughter, the princess. On the way, I will talk about the way afterwards, but on the way to solve the structure, we tried to find some starting models, and we thought about doing it from 2-dimensional sheets by electron microscopy, 3-dimensional reconstruction. It took us 4 months to get the first microcrystals. I'll come back to it. And 4.5 years to get these just monolayers. But once we found a way to do it, we could get them in 10 seconds. So these monolayers, when they were what is called in electron microscopy filtered, had the shape of the large subunit. We're talking here on the large subunit. And we could do reconstruction, which was not done before. And we saw something that we call the tunnel. So if you want to see, we saw something like this, and this is a tunnel. Until then, the tunnel was announced in the literature, or at least was suggested, first by Malkin and Rich, 20 years before we visualized it, based on biochemistry, and 3 years later based on more sophisticated biochemistry by Sabatini and Global. These 2 papers were very well filed in people's drawers or in garbage or I don't know where. It didn't get into textbooks, and I, being already 7 years in ribosome field, was not aware of it. But I had to convince myself that what I see here, you see it like from here or from top, that based on those that you saw a minute ago, I had to convince myself. I repeated this reconstruction 13 times, and then when I was sure, I went to the literature and I found out that there is a tunnel. So at least that I can see it. I didn't tell you yet, but when I started with ribosome crystallography, I was not well respected all over the world. Some people did, but most of them had questions or skepticism or worse about these studies. And coming out with, so I was Ada the village fool, or Ada the dreamer, or whatever you want to tell me now. I remember when I had the tunnel, I became Ada with the hole. And it took almost 9 years until it was rediscovered by cryo-electron microscopy. So now everybody knows there is a tunnel. I want to tell you, we could not convince the world that there is a tunnel, even when we saw it in the whole ribosome, not only like here in the large subunit. And for instance, in '88, just a year later, even Peter Moore, who is a well-known ribosomologist, doubted it. I don't really want to read it now, but there were lots of doubts. But there is a tunnel. You know now that there is a tunnel. Here is the large subunit. from the PTC to the end, this is the tunnel. We modeled into it polyalanine so we can see where it is and we can show in this, in this color orange. And I highlighted here part of the tunnel to show that somewhere there are places that it's very wide and others where there are constrictions. And we think that the very wild parts of the tunnel can host partially folded proteins, and based on some work that was published during the last, I think, decade and a half, that show that for hydrophobic proteins, there might be some beginning of folding within the ribosome. So this crevice, we think, could be the position where such folding starts. I don't know what will happen to such folding when it reaches this constriction here. And this is still open. I'm not even sure that there is folding, but we have some, I'll show you in a minute some evidence for that, where the constriction was cleverly chosen by antibiotics to block the tunnel, mainly macrolides, which is a very important antibiotic family that targets the ribosomes. So first of all, the crevice, we could bind a hydrophobic molecule into the crevice. And it's like a circular hydrophobic amino acids. And if one is ready to look together with me, so here is the modeled polyalanine, and that's the crevice. You see it's on the side. The protein can still go through the tunnel, at least until here. And something like this may happen, that instead of going straight, it can come here, fold a bit, and come back. So that's, I think, the only and still the most impressive possibility for ribosome to work as chaperoning, or to have some chaperoning properties. Let's look now at the constriction, where antibiotics bind. So because of the fundamental role played by the ribosome, a lot of antibiotics target it. Antibiotics, you know, this is the weapon one bacteria or one microorganism is developing against another microorganism. They don't do this for us, for using it in medicine, but in the middle of last century, it started, the use of it started. So antibiotics are like David and Goliath at least in our way of thinking. Bacterial ribosomes, those that are the target for antibiotics, are about 2.5 million Daltons. And antibiotics are about, most of them are about 800. They can reach 600 or 1,000. And the way they bind to the ribosome, or the way they stop the ribosome working, is more or less the way that the stone from David paralyzed Goliath. It hit it in an exposed position, which is important functionally. So this is the way the antibiotic works. It's not that just bind to the ribosome, but it has to bind to a functional site. And maybe I should say now, I should have said it earlier, all the functional sites of the ribosomes are RNA built, are solely RNA. PTC, it means where peptide bond is being made, also where the decoding is. RNA sites, means it's a ribozyme, and the antibiotics are mainly bound to RNA pieces. So here you can see 3 antibiotics that I used. I put, all what you see are crystallographic results. It means it's not just imagination. I put them into the structure of the large subunit where the RNA here is in bluish, and the proteins, all the proteins are in greenish. And the positions of the antibiotics, the magenta is at the active site, where the A site tRNA binds, the yellow is exactly where the peptide bond is made. I think you might have hit these names, not only the doctors, but also those that go just to pharmacy. Chloramphenicol, maybe less. It's not very useful. Clindamycin, maybe less because it's very, very potent. But erythromycin is a name that is more sold, and it sits at the entrance to the tunnel in the constriction here. Now, erythromycin is the mother or the grandmother of a whole family. So let's look at it, and maybe I'll say some words about it. Here is erythromycin looking into the tunnel. The ribosome here is in all types of green, RNA in dark, and proteins in light. And erythromycin here, sitting, you looking into the tunnel from top, and it blocks 2/3 of the tunnel. It means it just doesn't let the amino acid, the newly born protein go through it. From the side, it looks like that. So that's a slice of the large subunit in beige. T-site tRNA at the PTC. The newly born protein would have gone here if erythromycin does not stop it. So that's the way it functions. As I said, it's a mother of a whole family, or grandmother, which is called macrolides. And I really don't want to go a lot into chemistry, although it's a chemistry conference or chemistry lecture, but just to highlight that all macrolides are made of a ring. Erythromycin has 14 members, 2 sugars, 1 amino sugar and a normal sugar. Erythromycin is a good antibiotic, but it's not very stable in acid environments. And the stomach is an acid environment. So for treating problems in the stomach, companies try to make it more stable in acid environment. And they made 2 specific modifications in here and in here. Here and here. Here aliphatic, long aliphatic ring, and here just a methyl. That's not so important. The names of the results of the new compounds is also less important, but I will mention them. So let's look what happened just with these little changes. This is a view into the tunnel. You remember the arm that was jumping in, out is shown here. And now I'm showing here just the RNA now. I also didn't say that RNA occupies 2/3 of each ribosome, except for mitochondria, where part of the RNA is changed by proteins. But let's look into the tunnel entrance, and this is the way erythromycin binds. Now you see it atom by atom. Clarithromycin, which is just a little bit larger and more stable in acid, is even better occupying the void that is still available for proteins. And roxithromycin binds with two new anchors, and we dare to think that we see here structural basis for dosing. So